Hello, my name is Robert Dingley, and I'm presenting my poster on the regulation of catecholamine biosynthetic enzymes by the gas transmitters carbon monoxide and hydrogen sulfide. I have no conflicts of interest to declare. Increases in the expression of catecholamine biosynthetic genes and increased levels of catecholamines, specifically epinephrine, are found in patients with hypertension. The gas transmitters carbon monoxide and hydrogen sulfide have both been shown to have vasodilative effects. However, the role these gas transmitters play in regulating catecholamine biosynthesis and therefore hypertension has not been investigated. To investigate the role these gas transmitters play, we use the PC12 cell line derived from the rat adrenal gland. We treated the cells with the gas transmitters via donors that would release the gases into culture media. Carbon monoxide was supplied by the carbon monoxide releasing molecule 2, a ruthenium-based compound. Hydrogen sulfide was supplied by its solid analog, sodium hydrosulfide. mRNA was then harvested via trizol extraction, and the expression of catecholamine biosynthetic genes were measured via RT-qPCR. Figure 1 shows our carbon monoxide releasing molecule, or CORM, dose-response experiment. Cells were treated with doses of CORM ranging from 10 micromolar to 100 micromolar for 6 hours, followed by trizol extraction. We showed a relative decrease in the expression of two catecholamine biosynthetic genes, TH and DBH, with increasing dosage, statistically significant at 80 and 100 micromolar. Figure 2 shows our sodium hydrosulfide dose-response experiment. Cells were treated with doses of sodium hydrosulfide ranging from 20 micromolar to 300 micromolar for 6 hours, followed by trizol extraction. We did not see any difference in relative expression. We hypothesized that this could be the result of the hydrogen rapidly off-gassing from the culture media. In future work, we intend to investigate if the use of slow-release hydrogen sulfide donors or inducing endogenous production may have an effect on catecholamine biosynthetic genes. Catecholamine biosynthesis is regulated in part by glucocorticoids. To determine if our gas transmitters play a role in glucocorticoid-mediated expression of catecholamine biosynthetic genes, we treated cells with 1 micromolar dexamethasone, also known as DEX, a synthetic glucocorticoid, in combination with our gas transmitters. Figure 3 shows our DEX plus CORM experiments. We showed the expected increase in the expression of TH, DBH, and PNMT after treatment with DEX for 6 hours. However, a 15-minute pretreatment with 100 micromolar CORM greatly ameliorates this effect in all 3 genes. To determine what effects the gas transmitters have on actual production of catecholamines, we collected cell culture media from experiments for use in an enzyme-linked immunosorbent assay, or ELISA. This ELISA allowed us to determine concentrations of catecholamines in media of our cells for dopamine, norepinephrine, and epinephrine. However, PC12 cells do not produce epinephrine, so only the first 2 could be assayed. Figure 4 shows the results of our DEX plus CORM ELISA experiments. Cells were treated with either 1 micromolar DEX, 100 micromolar CORM, or both in combination for 24 hours. CORM alone had no significant effect on either dopamine or norepinephrine levels. DEX had the expected effect of increasing the relative concentration of both dopamine and norepinephrine. CORM in combination with DEX ameliorated DEX's relative increase in the concentration of norepinephrine. Surprisingly, CORM potentiated a DEX-mediated increase in dopamine. In conclusion, our work has shown that gas transmitters, particularly carbon monoxide, are interesting regulators of catecholamine biosynthesis. Future work will investigate the mechanisms by which this regulation occurs. Thank you for listening to this presentation.